Hello everybody, and thank you for watching this video on qualitative and quantitative analysis of pharmaceutical excipient. Today I'm going to talk about Polyquaternium-10. This video and many other videos in this channel are useful for people who are doing reverse engineering of pharmaceutical and cosmetic products. Usually I start the video with talking about the molecule a little bit. Then we move on and discuss if the material is compendial or not, and we discuss the IAD limits for the excipients, and then at the end we talk about the analytical methods for testing these excipients in the formulation. Now, Polyquaternium-10 is a catalytic polymer with the structure that we see in this slide. It has the amine group, and it has a polyether in the middle, and it has a polysaccharide at the right side of this structure. And the positively charged amine has a tendency to stick to the negatively charged hair. Polyquaternium is a nomenclature given by INCI to at least 3 different type of polymers. And these polymers they all share the amine, positively charged amine group, but chemically they are all different. So the Polyquaternium-5 and Polyquaternium-7, they are completely different molecule, and it is different than let's say PEG 200. That the difference between PEG 200 and 300 is just the average molecular weight. In the case of PEGs, they are all the same molecule with different polymeric chain, whereas in the case of Polyquaternium, they are just completely different chemicals. These materials are not compendial raw materials, so they are not listed in USP, EP, BP, or any of these publications. The IAD database, there is only one entry for Polyquaternium-10, and it is for a topical shampoo with a maximum limit of 2%. There is an entry for Polyquaternium-7, but this is completely different molecule. Well, it is for information that for case of Polyquaternium-7, the limit is 1%. So there is one approved drug product that contains Polyquaternium-10 in the formulation with a maximum limit of 2%. So for people who do reverse engineering, they have to keep in mind that in their formulation that they are working on, they cannot have this excipient more than 2% weight per rate. For qualitative and quantitative analysis, I will describe 2 methods. Both are with the HPLC, one with UV detector, the other one with ELC detector. This is the chromatogram generated by HPLC ELC detector. The column is C18, and it is gradient program. Mobile phase A is water acetonitrile with TFA, water B is 100% acetonitrile. Diluent is a water with TFA. The interesting thing about this chromatogram is that there are 2 peaks associated with Polyquaternium-10. One of them is at 2.2 minutes, which I call it Polyquaternium-free Polyquaternium, and the other one at almost 5.25 minutes, which is I call it Polyquaternium complex. It turns out that Polyquaternium has a tendency to create these the ion pairing agents or complexes with any negatively charged molecule or excipient in the formulation. So if you have negatively charged excipient in the formulation, which is quite possible, then you will form a complex. And so this was you have to keep in mind that when you're doing reverse engineering, you have to make sure that you include all these peaks associated with polyquaternium-10. In this case, the chromatogram that we're looking at, let's say if someone overlook the peak at the 5.25 minute and they only focus on the free polyquaternium-10, you can imagine that your amount determination would be extremely small, and maybe only 5% of the true value. The second thing that I would like to talk about is that the polyquaternium-10 is a rather difficult molecule to work with, and you should avoid using glassware. And this material has a tendency to stick to the glass, and to clean it off, you really have to use very strong acid, like concentrated chromium acid, to get it off. It is a difficult molecule to work with. So if you could use plastic rather than glass. But if you use glass, keep in mind that you have to do a lot of acid wash. The next method is HPLC with the UV detector. Now the condition here are the column was a mixed mode column, that you had the hydrophobic and hydrophilic stationary phase. Mobile phase is as phosphate acetonitrile, the isocratic. Detection is done at 210 nanometer, and water is diluent is water. In this chromatogram, we only see a single peak. There are two peaks here in this chromatogram. The first one is actually all placebo related, meaning that it is the other excipient present in the formulation. And the peak near 2.5 minutes is the polyquaternium-10. There is no other detectable peak associated with polyquaternium-10. It is possible that in this second formulation is either polyquaternium did not make a complex, or if it did, both peaks are eluting at the same retention time. So with the two methods that I show you, you would be able to do the quantitation, meaning that you can do the amount determination, how much is in there. But in both chromatogram, there were no profile that you could make a grade determination or pinpoint who was the manufacturer of this raw material. There was no other information than a single peak. The second important point that I want to just to repeat is that you have to keep in mind the polyquaternium-10 has a tendency to make ion pairing, the negatively charged excipient in the formulation. So you have to keep an eye on those possibilities. The references is just FDA or the supplier's website that they are providing columns for the quantitation of excipients. Thank you very much for watching this video, and have a great time.